Bismillah ar-Rahman Rahim. Now this is another paper 6 which we're going to do for the 5090 Biology O-Level June 2020, and I'm going to discuss this and help you with the exam skills that you must have. The first thing to remember is that this paper is for 1 hour and you must be very careful about the time that you are going to spend on each question. You must read firstly the whole question and then come back and start attempting it. That's the first rule that you follow. Number one, you look at the time. You keep a track of the time. You must wear a watch or something. Number two, read the whole question. Read the first question, the whole of it. I might not do this in the recordings because I have to do it according to a certain other rule that I follow. So, but these are the rules. You see, what problem that I see you all are facing, you put a lot of comments, and I've gathered something is that when you do a paper, you don't learn the skill of doing the paper. You just sort of memorize that paper and those questions. No, that's not the way. You should think, how am I going to do the paper 6? And what are the mistakes that I make and how I'm going to improve that. So you are not really working on your skill. You're not working on your exam skill. Basically, you might have done a lot of studies the whole year round or in the 3 years that you're taking your O levels. But what matters is that in this 1 hour, what did you do? How well did you do the exam? And how many mistakes did you make? First thing, there is no choice in this paper. You have to do the whole paper. A student used the apparatus shown below to investigate respiration in some small insects. The apparatus was sealed with a clip so that any changes in the volume of gas in the apparatus caused the bubble of colored liquid in the capillary tube to move. So this is the bubble of colored liquid here. So I'm just going to color that red. And this is going to move. They just said move, right? Then you have a clip here. Look at it carefully. Glass tubing, small insects, metal gauze, chemical which absorbs carbon dioxide, capillary tube, scale, and a rubber tubing, and a clip. Then there's a bubble of colored liquid. So you look at this carefully and see what are we studying? We're studying respiration. Respiration means what? The release of energy using food in living organisms. So glucose is respired to release energy. Now let's look at the first question. It says suggest what happens to the bubble of colored liquid as the insects in the test tube respire. Now basically what you have to understand is the insects are respiring. Please understand, respiration is something else, breathing is something else. When they're respiring, they're using up oxygen. So they're taking up the oxygen in this thing, but then they're also producing carbon dioxide. So the carbon dioxide is going in here. The carbon dioxide is going to be absorbed by this chemical. So they're taking in oxygen. So say if there were 100 molecules of oxygen here, now there are 50 left. Now that is going to create a pressure difference. And this colored liquid is going to move this side towards the insect. So this is what you have to understand, is that a pressure difference is being created. Just like if you have a glass and you have some liquid in it and there's a straw, why would the water move up and why would this level go down? Is because the water is being sucked up by somebody and it's going into his mouth and then into his stomach. So you've got to understand there's this suction force being created. But why is the suction force being created? It's a pressure difference which has caused this. So the bubble of colored liquid is going to move where? It can't move this side. No, it can't move this side. But what if something was being created here and pressure was created from this side? Then only it would move this side. So understand when it would move this side and when it would move the other side. So moves towards the insect, or you can say to the left. Using the apparatus in the diagram, the student read the starting position of the bubble of colored liquid. She recorded the reading from the left edge of the bubble, left edge of the bubble. She left the apparatus for 60 minutes and then recorded the final position of the bubble. The diagrams show the starting and the final position of the bubble. In the spaces provided, record the reading for the position of each bubble. Now pause the video here, have a look at it. I'm not writing the answer as yet, and I want you to figure that out and then look at my answer later on. So you need to pause this video. When you're watching this, you must wait and do it yourself, or you should have done the paper first and then you should look at this video. So you can see the answer to 6.8 and 5.6, and this was for 2 marks. So please remember, you had to get 6.8, it says in the space provided, record the reading for the position. Then coming to part 3, it said calculate the distance the bubble moved in 60 minutes. So that was easy because you had to just minus this 6.8 minus 5.6, and this was 1.2. But look at the 2 marks. What is the 2 marks? One is for the unit. Now it is so sad if you didn't write the unit and you missed this 1 mark. So 1.2 is 1 mark and centimeter is the second mark. Then part 4, calculate the rate of movement of the bubble in millimeter per minute, and spaces for the working. So you have to again look at it. This is now 2 marks, but the reading has to be in rate of the movement of the bubble in millimeter per minute. Now as you can see, it was millimeter, so 1.2 centimeter is equal to 12 millimeter, and then 12 divided by 60, because this was in 60 minutes. Here was 60 minutes, if you didn't see that. And so it will be 0.2. So for 2 marks, these are the working. There was 1 mark for this and there was 1 mark for this. Then part B of the question, part 1, explain why carrying out the experiment several times would improve the reliability of the results. You see, I always say, remember, R for reliability, R for repeat. So reliability means that the tests that you did were very reliable. Reliable means that your results were very accurate, not accurate. Reliable is a different thing and accuracy is a different thing. Means that this would be a result which will go across the board and we know that this is the final result. So why do we repeat the experiment is that it says why the experiment several times would improve the reliability of the main thing is that then you can calculate the mean. When you have 5 readings or 3 readings or several readings, then you can allow the mean to be calculated, allows mean to be calculated. You see, remember, if you get a reading which is 10, and then you get a reading 11, and then you get a reading 20, and then you get 11, and then you get 12. So you know this is the anomalous reading. This is the odd reading in which this is not correct, because you see all these reactions which are taking place are due to enzymes. And enzymes work best. They can't suddenly go fast forward or they go into slow motion. This is not possible. Enzymes are biological molecules and they always work best at an optimum temperature and pH. So how can you get a reading? If I say let's measure the rate of enzyme reaction and it's in bubbles or something, so it's 10 bubbles, then 11, and then suddenly there are 20 bubbles. So this is not very reliable. Like for instance, give you another example. You get a reading 10, then 11, then you get 9, and then you get 20, and then you get 21. Well, then you know either these one are correct or these one are correct. So you need to do another 2 experiments, and the next reading you get is 10, and then you get 8. So you realize these were the anomalous readings. So when you take several readings, you can then find out and identify the anomalies. This is called identify anomalies. Identify anomalies. Anomalies means the ones which is not, or you can say the odd results. Like for instance, I give you a very simple example is that I gave you a bio exam and I've been teaching you for 2 years. And usually the students get 50% marks, one of the students. And this student always gets nearly 50, 55, 45, something like this. And in one test, he gets 90% marks. And then you realize, my God, what's gone wrong? Of course, we know what's gone wrong, but then that doesn't tally with his normal trend. And then, of course, some of you will say, no, miss, he worked very hard. Well, I'm glad if he's worked very hard, but then I'll do another test. And in the other test, if he still gets 55 marks, 55% marks, then that means that 90% was just because maybe he had some help in some way. Then coming to part 2 of the question, it says suggest a suitable control. I always tell you control is for comparison. You say, okay, well, these insects are respiring, so this dye is moving or this liquid is moving. Now I can say, okay, no, it will move even if there are no insects. So then all my theory is gone wrong. So then what do? Either we put in, what do we do? So control is, you always do is you remove that organism which is actively working or which is the biological organism which is causing those reactions to take place, and then something is happening. So we use the same apparatus but with no insects. And if it still moves, then it means it wasn't the insects which was causing it. So you can either use dead insects, but then you'll have to see that there are no microorganisms on that, because dead insects would have bacteria and fungi on them. So you'd have to either sterilize them, or you'll have to sort of boil them, because there should be no microorganisms on them. Or you can just put some glass beads of the same volume. Remember that, of the same volume. So how would we word it? Same apparatus. This is important. Same apparatus is important with dead or no insects or glass beads. Then part C1. Suggest an environmental factor outside the apparatus that might affect what happens to the bubble of colored liquid. What would happen? The temperature. If this temperature in this room is very hot, or the temperature where you're doing this is very cold, you've got an air conditioner working and it's very, very cold. So then, of course, you've got to have this. So temperature would be the thing, environmental factor outside the apparatus that might affect what happens to the bubble. Yes, because please remember, respiration is the reaction which requires enzymes. And you know enzymes are affected by temperature. So for 1 mark, that was all what you were asked. Then it says describe how the student might ensure that this above factor, has no effect on the bubble of colored liquid. So what would you do? You would use a thermostatically controlled water bath, because you would use a thermostatically controlled water bath. I always say there are certain sentences that you need to remember. Don't write anything else. Well, a student wrote bubble bath. I said, wow, that's nice. Use a thermostatically controlled water bath. And then when you tell the student you wrote bubble bath, he says, well, miss, I meant water bath. Well, you can just do the correction. I'm sorry, examiner is not going to do that correction, that you wrote bubble bath, and I'm going to say, okay, he meant a water bath. So use a thermostatically controlled water bath would get you that 1 mark. Question D1. The insects produce carbon dioxide as a result of respiration. Suggest and explain what would happen to the bubble of colored liquid if the experiment was repeated with the chemical which absorbs carbon dioxide removed from the apparatus. So let's look at the apparatus again. Now, if you look at the apparatus, this is the apparatus. Now, if we remove this chemical, let's remove this chemical, right? So we've removed this chemical. Now there's no chemical here, and we have just this test tube and no chemical is here. Now what is going to happen is it's going to take in oxygen and it is going to produce carbon dioxide, but there is no chemical here. So this bubble is not going to move. Why is it not going to move? Remember your equation for respiration. It is C6H12O6 plus 6O2. And what's it to give you? 6CO2. So if it is taking in 6 oxygen, it is also producing 6 carbon dioxide. So this bubble is not going to move. And of course it is 6H2O, but I'm not talking about that. But what I'm talking about is this. It is taking in 6 oxygen and producing, so this bubble is not going to move. It's going to stay here. This is not going to move. So just look at it. How did we word this? So the 1 mark was bubble does not move. This got you 1 mark. And the second mark was the volume of CO2 produced and the oxygen used up is in balance, so it is not going to move. Now, coming to the next part of the question, it says the student wanted to show the insects produce carbon dioxide. The diagram shows the apparatus she used. When carbon dioxide is bubbled through lime water, it turns out clear. It turns from clear to cloudy. Now you can see there's an air in, there's air out. Very important that you see is this is dipping into it, this is going out, and then this is going in. Which dips in? This is now going in, dipping in. This is very important that you note this. Test tube A, test tube B, and test tube C. So this is the outside air which is entering this first tube, and then the air is going into this tube where the insects are lying, and then the air is going to go into this. So when carbon dioxide is bubbled through lime water, it turns from clear to cloudy. They've given you suggest and explain the results you would expect after the apparatus was left for 30 minutes. So this is the outside air which is coming in here. And this is the air which is coming from the insects which is coming in here. Of course, it's coming through this. I'm not saying it's coming through this one. So what will happen in test tube A? Test tube A is the outside air. So lime water remains clear because the outside air does not contain so much carbon dioxide. It contains very little carbon dioxide. Now test tube C, now here the lime water is going to turn milky. Why? Because the insects are producing carbon dioxide. And that air is going here and then coming here and passing through the lime water. So that was the explanation for it. Suggest and explain the results. Here we have the outside air. The outside air is very little carbon dioxide, so this will remain clear, or you can even say it can become slightly cloudy. But this one is going to be turned, this one is going to be cloudy here. Why? Because the insects are producing carbon dioxide and all that is going into test tube C. Now I'm wording it for you how we'd word it, because some of you maybe know it but you didn't word it correctly. Please remember the wordings are very important. The biological English has to be very exact. So test tube A, lime water remains clear. Atmospheric air contains very little carbon dioxide. Test tube C, lime water turns cloudy. Why? Because there's more carbon dioxide into C which has been produced by the insects. Now coming to question number 2, the photograph shows the cut surface of a lemon fruit. In the space below, make a large drawing of the cut surface of the lemon as it appears in the photograph. So please remember there are 4 marks for this. And everyone, all of you say that, oh, miss, we can't draw and all this, but we don't expect you to draw. We just expect you to draw what you see. So I'm sure even if you're the worst at drawing, you can do a good job of it and get your 4 marks. Now I know this diagram might not be very exact, but this is all what you have to show. Clear and clean lines drawn with a sharp pencil. You shouldn't be having this sort of this shaded lines. You shouldn't be doing this. That shouldn't be there anymore. So clear and clean lines, and that got you 1 mark for that. So clear and clean lines drawn with a sharp pencil got you 1 mark. Detail a boundary between pith, peel, and flesh. So the pith, peel, and flesh, you should have shown it with these 3 lines which I've shown you. Then the segment details you should have given me, and of course there's a minimum size. Segment details you can give me a little more as well. Maybe you could give me a bit more of the segment details, but then you'd have to be exact how many segments were there in it. I've just done a few of them, but you can of course look at it carefully. I always say is that you should always just very easily just shade it. This is the lines that we have expected you to, the line, then another line, then these segment details. So just like if you are tracing it, but you have to make it a little bigger. And then this is the area that you have to draw. So if you just see that you are actually tracing it. So this is the first line. I'm not able to do it because I have a very thick marker, and then this is the second line, and then of course this is the third line which you have to give me. So I'm sure all of you can do these drawings. If there's anything which is a problem for you, please do comment and I'll try my best to explain it to you in another video. Now it says on the photograph, draw a straight line between A and B. Measure and record the length of this line. So on which photograph is it saying? This is the photograph. So you have to draw a line between A and B. This is what you have to do on the photograph. It didn't say on the diagram because it says here the photograph. So this is the photograph. This is where you would make a mistake. I know it. I'm 100%. So it says on the photograph, draw a straight line, measure and record the length of this line. On the actual lemon that was photographed, the measure between A and B is 6 centimeter. 6 centimeter is equal to 60 millimeter. This is the first thing you do. Use your measurements in B1. Use this measurement to calculate the magnification of the lemon in the photograph, and this is a space for working. Now, you measured it. I'm not sure what the measurement is. I would say it was 12 centimeters, so it would be 120 millimeter. Now, on the actual lemon that was photographed, the measurement is 6 centimeters, so that's 60 millimeter. Now, space for working. What is the formula? Magnification is equal to image over actual. This is the formula that we use all the time. So the image on the photograph was 120. The actual is 60. So what is the magnification? It is x2, because this would be is equal to 2. 60 divided by 120 divided by 60 is 2. So the magnification would be 2. We always write magnification x2, but the x has already been written for you. So the 3 marks were for what? The 3 marks for you to convert this into millimeter. 1 mark is for that. The correct formula got you another mark, and then the correct answer got you another mark. Then coming to the C part of the question, a student tested some juice from the lemon for the presence of reducing sugar and protein. Name the testing reagents she used for these tests. For reducing sugar, what will it be? Very basic. For the reducing sugar, it will be Benedict's. And for the protein, it will be biuret. So Benedict's and this would be biuret. So you had to know the name and you just had to write that, and that got you your 2 marks. Now it says state an approximate temperature at which each test should be done. Now any temperature sort of in reducing sugar test, it can be anything above 80 degrees Celsius, 80 to 100 degrees Celsius. That is the normal that we follow. So anything above 60 was formed. And in this, of course, any you could have said room temperature also. In this, we have no temperature story here. Or you could have said anything below 40. So you could have said 30 degrees Celsius. Anything below 40 was fine, right? Then coming to the part 3, from the results of a test, the student concluded that reducing sugar was present in the lemon juice, but protein was not present. State the results she observed for the test that helped her to come to this conclusion. Now, the reducing sugar could have been anything. You could have said anything because you see, we don't know. There are different values. If you got less sugar in red, reducing sugar, it would be green. If it would be more, it would be yellow. So it could be green, it could be yellow. What else could it be? It could be orange. So we could have an orange one as well. And then of course we could also have it red or a brick red color. So any of these colors would have proved that a reducing sugar is present. Now coming to the protein test, the protein test will remain blue. We don't have to write remained, but because you see the biuret reagent is blue, so it remained blue. Now this was for you too, for the 2 marks that you were supposed to give me. Then coming to the part 4, the student then tested the lemon juice for the presence of fats. Explain why she ensured that there were no naked flames in the laboratory when she did this. Why? Because ethanol, you're going to use ethanol. And ethanol is highly flammable. So for 2 marks, you had to say is because using ethanol or alcohol, you could have used alcohol as well. And this alcohol is highly inflammable, so it can catch fire, or you can say it can catch fire. So you can have said this. Part D of the question, another student investigated the concentration of reducing sugar in lemon juice using a different method. First, he tested solutions of different concentrations of reducing sugar glucose. He used an instrument called a calorimeter to measure the concentration of glucose. The lower the number on the calorimeter reading, the higher the glucose concentration. His results are shown in the table. Glucose concentration in percentage and calorimeter reading in arbitrary units. On the grid, construct a line graph to show the relationship between glucose concentration and calorimeter readings. Join your points with a ruled straight line. Now this is a 4 marks which nobody should miss out. This doesn't need any biology. You don't need to know any information. You don't know any bio. That's fine. The rule is what? The rule is that this is the first column. The first column is always the independent variable and it is plotted on the x-axis. So this whole story, glucose concentration percentage, must go on the x-axis and this must go on the y-axis. This is the rule that I ask you to remember. The first column is always the independent variable. You study this in A levels if you do A levels. So the first column is always the independent variable and that is plotted on the x-axis and the other will go on the y-axis. So on the x-axis you have to write this. So if you just wrote this and you didn't draw any graph, well, you got 1 mark. For not drawing any graph, just labeling the axis, glucose concentration, percentage, giving me the units here, very important, giving me the units here, arbitrary units, you had to write that. Just doing this, you got 1 mark for this. Now you choose the scale very carefully because you must use the grid, the entire grid. You must use maximum part of the grid. So I've used nearly, there's only one little box left which I have not been able to use. So I've decided to use this and then you plot the dots and then you connect the dots point to point with a ruler. You connect the points point to point with a ruler. Please remember, you do not extrapolate. Extrapolate means you do not extend it. You only connect the plots that were given to you on the before in the columns that you were given. You were only supposed to plot these and then you are supposed to connect them. You're not supposed to extend them. Next part of it is part 2. The student then tested some lemon juice and obtained a calorimeter reading of 0.65. Arbitrary units. Use a graph to find the concentration of glucose in the lemon juice. Show you're working on the graph. So you have to show this. This is for 2 marks. So you have to show me the working and you have to show this on the graph, and then you have to give me the reading. The unit has been given to you. So what are the 2 marks? 1 mark is for you showing it to me on the graph, working shown on the graph, and the correct value from the graph. Now, what you have to understand is that they said 0.65 arbitrary units. So that would be somewhere here. So then you draw a line here and you see where it cuts it, and then you go right down and read off the glucose concentration. Now, whatever that is, you have to give me that figure here. Whatever it comes to, it's between zero point 0.5 and 0.75, and it will be, so you figure that out and then you write that here. So, but the main mark was for you to show me this here, this green line which I've shown you. You have to show it to me here, and then you have to give me the reading here, which was what you will see from the graph. And so, of course, every student was different because whatever you read from the graph was correct. It says working shown on the graph and the correct value from the graph. So you have to give me the correct value, whatever was your value from your graph. Thank you very much for watching, and please do subscribe and do give a comment of anything else you want me to do. And I hope this is going to help you in your future coming exam. The exams are very near, and wish you all the very best. Thank you very much.